back. I hope that you've all gotten something out of day one and enjoyed it. We're going to be sending around an assessment sheet. So assess us. We want to improve. So tell us what was good and what was bad. And we want to thank all our pals for coming to the CRLI and wanting to be advocates. You really inspire all of us. I know I'm speaking for the entire faculty. We wouldn't want to be anywhere else on a Saturday morning, believe it or not. And, so, we really appreciate it. And today what we're going to do, it's going to be a nice day. I'm going to tell you how to read a research paper. You're going to look at a paper and really don't get overwhelmed. It's actually, really going to be made very easy and straightforward. We'll have small groups that go over it, and then we'll actually go over the answers. So do not sweat, but we want to give you some tools. So when you read something, you read it critically. And then we're going to talk about some opportunities and share some ways that you can advocate. Lyle's going to tell you a little bit about this congressionally directed military research program and how you can be active there. We have Todd Kelly, who actually is a CRLI graduate who participated in the review process. Gary and Jamie are going to tell you a little bit about their experiences with group advocacy. And then Rick is going to tell you how to get involved and easy ways to get involved and some ideas. And then after that, Dr. Ellman's going to lead a roundtable where we're all going to talk about what are some of the things that you've thought about that maybe ways you could advocate. The faculty will give you some ideas, but we want to hear what you're thinking after all of this education. And then we'll adjourn, and you guys will be research ambassadors, and Sarah will be sending out your certificates. So that's what the day looks like. We're going to start by asking everyone to show their faces, because we're going to take a group picture. Unless, if you feel strongly about not being in the picture, that's fine. We understand. But believe me, I probably feel the strongest about this aspect of things. But we're going to, that way we can sort of tweet it out and everybody can see that this was an active group. And we're really proud of our CRLI graduates. So Sarah, you're doing it. You tell us when, and then.
All right, give me 1 second.
Can't smile for too long.
Yeah, that's right. My face will freeze.
Okay, ready? All right, hold on. One more. Looks like Noah's trying to join. Let's get him in here.
I see Jack. Jack joined. Morning, Jack.
All right, Noah, you ready for a picture?
Let's go.
You don't have any hair, Noah, to fix.
Oh yeah, that's why I like it.
All right. Wait, hold on. Nope, not wrong. Close. Too many buttons. Ready and go. Cool. Awesome. Thanks, guys.
Okay.
All right.
So hopefully everybody is ready to go. So can everybody see my screen?
Yeah, that's good.
Yeah, everybody see my screen?
Yes.
Okay, good. Because I can't, boy. Well, okay. So today we're going to learn how to read critically. And so you really don't have to be a scientist. You just have to use some some systematic way to approach a paper. So, oh boy, let's see how we move it. There we go. So here's what we're going to talk about. First, I'm going to give you some general pointers, just sort of general things to think about. We're going to talk about what are the sections of a research paper. And then I'm going to give you some step-by-step ways to go through a paper. And understand that some of this is going to be repetitious, but sometimes repetition reinforces. So first, think critically. Don't always think that because somebody has an MD, a PhD, or whatever after their name, they know everything. Don't always assume that authors are correct. Ask questions, and think of questions in your mind that you would have and see if they're answered as you read through the paper. And think about, are the investigators actually asking the right questions, or are they solving the right problems, and are they approaching it logically? Are the assumptions that they make reasonable in your mind? And read creatively. What do you think was good about the paper? What do you think was sort of novel and interesting? And is there translation of those ideas elsewhere? Don't be embarrassed to take notes. If you saw some of my college textbooks, I mean, yeah, they're just overlined. Everything's yellow, and there's all kinds of notes in the margins. Always think about questions and criticisms. Underline what you think are the most important takeaways. What's the most important thing about what you read? And mark what you think is questionable also. So, and after you read anything, think about it, and can you actually summarize it and teach it? Everybody always says, if you can make something simple to someone else, you understand it. And if you, after you read something, you can sort of put it together in a few sentences, that solidifies it and underscores that you understand the material. And there may be, that way you may see, hey, I better go back and read that again. I didn't quite understand it. You can always consider, if you're really interested in a topic, going back and looking at the background, the references. And always think about, what did this study add? Was this just a me too? Or did it really grow and enlarge our knowledge base and extend it? And it's going to be slow at first, but don't worry about it. I mean, that's the way things are. You're not going to be like, oh, great, from the start. So now, what are the sections? So those are some pointers as you go through a paper, just keeping those in mind. But let's talk more solidly about what can you expect if you're opening up a research paper. You heard about this clinical trial and you wanted to pull up the actual paper and read it. What are the sections you're going to be facing, and what do each of them contain? There's going to be an abstract, an introduction, methods, results, a conclusion, and then of course the references that the work was based on. And let's start with, what's an abstract? That's actually the summary of the paper. It should maybe give a little bit of background that led to what the question was. It should present the key results, really what they want your take home to be. And then it's going to give you a brief bullet of what you can conclude from the results. And so, some people go right to that, but as I'll show you when we talk about how you read a paper, you read the abstract last. So let's start with the introduction of a paper. So we're opening our paper, and the first thing we do is look at the introduction, and that's going to introduce the background. What's gone before? What led to this question being asked? And it should have the previous work, and it should be a very logical extension that, because of this background, this is why I'm asking this question. And it should really outline the basic reason for the study and the question being asked. You should be asking, before you even finish, you should be saying, well, somebody should look at this. The methods clearly tell, okay, I've got this question. How am I going to answer it? What am I going to do? How am I going to design a study that answers it? How am I going to carry out the experiment? And then the results are the results. What did we find? What was the data that we got from this experiment? And the conclusions should just be, okay, I have these results. How do I interpret them, especially in the context of what we know, especially in the background? And that means you have to, again, go in what previous work has been done in the area. How do your results fit into that and extend it? And where do we go from here? What should the next steps be to enhance our knowledge base and to move the field forward? And then, of course, the references simply are the background and the literature that your study was based on, or that the study was based on. So moving to the next step, okay, we know the parts, we know the sections of a paper. We have some clues about what we should be doing when we read a paper, some sort of ideas. But how do we step by step read a paper? Well, first of all, when you read, look up words you don't know. And I always say Google and Wikipedia are your friend. Just Google it. I mean, God, what do we do? I don't know. I'm sure, well, I'm pretty old, but I remember going, I wanted to do research or look up references on a topic. I had to go to something called Index Medicus, which weighed as much as I do, each tome, and it would be all the publications for a whole year. And I'd have to go through that and look it up by area of interest. You know, maybe it would be pathogenesis of ALS or whatever, or oxidative stress ALS. And it was just so tedious. Now I just Google it. And if I don't know something and I want to read just a summary about a topic and I Google it and I go to Wikipedia, I'll get a whole, you know, sort of summary about that area. So, I mean, in the past, this is so different than what we were used to 20 years ago. So Google and Wikipedia are your friend. Take notes when you read and reread. If you're not sure, just reread the section. Start. Now, here's how we start. We start with the introduction because that lays the foundation, the groundwork, gives you the background. It's sort of like the foundation you're going to build the house on. So it presents the supporting information on which the study was based. And it then should lead right to what the big question is, what the problem is that the field is trying to solve and that this publication or this experiment was trying to look at. And it should really reinforce the importance. And it really should be so logical that this is the question. It should lead directly to the specific question. That question shouldn't come out of the blue. It should be built on, you know, A happened, then equals B. Oh, what happens is B? Does it lead to C? So it should lead directly to the question being asked. And you want it to sound logical, you know, and most of you can think logically to some extent. So make sure that after you read the introduction, you really can summarize the background and what's being asked and what the results are going to add to the field. Then you read the methods and you want to look at the approach. Now, this can be really tough for you guys on a, you know, basic science paper. So, you know, you want to get the gist though of how are the investigators approaching? Does it seem logical? Is the design of the study likely to be valid? Will it answer the question? You can even try making a diagram, use that pen and make notes and, you know, diagram what the investigators are doing. And, you know, what I think many of you are going to be most interested in are clinical trials. So what you want to do when you look at that method section is, how did they design the study and control population? And our paper is going to give you some clues, and you'll see that it isn't so difficult. You know, how was the medication dosed? How did they judge safety? How were the treatment and control groups determined? How is efficacy measured? And thanks to Eric, you'll now know how the results were analyzed, right? Everybody knows a p-value by now, right? But you know, you can see, did it seem to be a good analysis, a valid way to look at the results? And then you read the results. Pay attention to the figures and tables because they often contain the most key points that the investigator wants to make. Pay attention to whether the results there answered the question that was being asked. And in clinical trials, were the control and treatment groups similar at the start? Were there safety issues? Did the intervention actually work? Was there statistical significance? So, and you know how to do that now. And then you read the conclusion, discussion, interpretation. It's called different things, but basically those results, how does it fit into the grand scheme of things? And what does it mean for the field? So did the investigator interpret his results correctly in your mind? Do you agree with it? Is there any other way that those results could be interpreted? And does the investigator also talk about weaknesses? There's always weaknesses. There's always problems. And if somebody doesn't admit to them, then there may be a problem. And does the investigator take the results that they have in their conclusions and put it in the grand scheme of things? And always, what does the study add to what we know? And where do we go from here? And references they're there for you to look at if you want more information and you want to really know about the background. Now you go back to the abstract. And was that an accurate summary of what you just read? So, to end, do not be afraid to jump in and read the literature. Be an informed consumer. That's really important. Learn to be critical and learn to be creative. Ask questions. Let me tell you, some of the questions folks ask, pals have asked me, I don't know the answer to. They ask really good questions. You have the ability to ask really good questions and prompt us to really think. And, with practice, this will be much easier. So we're going to move into, does anybody have any questions?
Terry, this is Rick. I'll just make one comment. It's a great presentation. I think it's so important that we try to read the original paper. So often now what we're reading is someone else's summary of the paper in social media or, on a website. Oftentimes that summary is not accurate. We have to go back and read the paper ourselves to really understand it.
Exactly. And, if you don't understand it, bring the paper in. I've had people bring papers in and ask me to look at it. And what does this mean? I saw this, and don't be afraid to ask, if when you bring it into your clinic doctor, they don't want to talk about it or they're uncomfortable, well, that would be a concern to me. I don't know how the other faculty feel, but, bring it in and talk to us about it.
Remember what I said in the beginning too. Faculty of the CRLI, we're here to mentor you for as long as you want us. So if you have something that you don't understand, we're happy to have you email us, send us the paper. If you want us to jump on a quick Zoom call with you, I'm more than willing to do that.
So we are going to now go into breakout rooms, and we're gonna answer these questions. Does everybody have their sort of small group exercise sheet handy? I hope. So Sarah, if you could put us in our breakout rooms, then we'll start to work on this, and then we'll come back and I'm going to go through all the answers with you. And you'll see we're going to use some of the concepts that we've been talking about in this talk.
Journal, for example, the editor would have taken a look at it and sent it back the next day and said, okay, not for our journal.
So,
Or they would have encouraged them to report on features that got omitted. I'm not sure that it's an unreportable study. It probably wouldn't get into any journal. You're right.
No, I agree with you, Eric. That's what I was saying before, is I applaud anyone who's out there trying and publishing. But, this almost certainly wouldn't have got into a journal with a really high impact factor. It doesn't mean it shouldn't have been published. It should have been published somewhere, but with a lot of changes.
So I hope everybody had a good discussion. We couldn't even get through half the questions because our discussion got so lively. But let's go through the answers so we're all on the same page. I'm going to share my screen here.
I'll ask you about the thing about that.
What was that? Let's see what happens here. Okay, can everybody see my screen?
Yes.
Yes.
Okay, good. And tell me if it moves along. So here are the questions, and I've outlined on the actual questions where you find the answers. And that's, of course, in your handouts, you have these slides, but let's go through. How many people were enrolled? Anybody want to yell it out? Yeah, there we go. We screened 32 folks, 23 were enrolled, 10 were treated. There were 13 controls, 3 of whom got lost, so it was 10 and 10. How long was the trial? Feel free to yell out the answers.
1 year.
1 year. Was there a sugar pill or sham surgery used?
No.
Was the study blinded? In other words, did the investigators know who was in the treatment group and who was in the control group or not? Well, the study wasn't blinded. And of course, best way to do a study is pretty much to be a double blinded where neither the participant or the investigator knows what group they're in. How did they measure safety and efficacy of the stem cell treatment? And this was in the methods analysis. Well, for safety, there weren't any clear plans. Now, this is really bad, right? You need to have a safety monitoring. We have data and safety monitoring boards in our clinical trials typically. So there were really no plans and there was no safety data presented in the results section. And how about efficacy? What data points? Well, they looked at survival. They did follow the functional rating score, ALS-FRSR, the vital capacity and a quality of life. Now, the inclusion and exclusion, this is who could go in the trial and who were excluded. And so, of course, they had a list. If you look in the method section, you had to have ALS, you had to have negative MRIs, you had to have certain FVC values and an appropriate nutritional state. And what excluded you was if you had really bad bulbar involvement, an inadequate nutritional state, tracheostomy or gastrostomy, or other systemic disorders that would be overwhelming, and any evidence of disease on your MRIs, what we call spondylitic myelopathy, which means pressure on the spinal cord from changes in the spine. So how were the treatment and control groups chosen? Anybody want to comment on that? Well, it was just done by the site investigator. There was no randomization. And I think Eric can talk to the problem with not doing something randomly and trying to analyze it. And the way controls were selected was just by saying, I don't want that. So, it's really not a good way to develop your treatment and control groups. And so how about if we compare the two groups then? Okay, so they were self-selected. How did they compare in terms of duration of the ALS disease? And unfortunately, rather than from symptom onset, it was from diagnosis to baseline. How about their functional rating score at baseline, their severity at baseline. And then when you look at this, were these two groups similar? So let's start with duration from the diagnosis to baseline visit. Anybody want to yell that one out? No. Well, if we look at this at the table, and remember we talked about looking at the tables and figures when we got started. If you looked at the duration of disease between the control group and the treatment group, what do you notice? Much longer in the treatment group. So in the treatment group, 30 months, much slower disease to begin with, compared to the control group where it was only 14 months. What about the scores at baseline? Again, we go to our table and here's our baseline ALSFRS score. If we look at that, and then we actually have our comparative, our graphic, you can see again that there was a huge difference between the two groups, with the control group having a much higher ALSFRSR than the treatment group. They had been going on longer, progressed, but at a slower rate. So what do you think? Who would think that the treatment groups and the control groups were similar? They were very different. So again, you're comparing apples and oranges. And so these are some of the problems. And, you know, I hope that some of the groups got through these questions so that you can see that you were able to look at this critically, even though, you know, you're a novice at it, or maybe you haven't read as much. What percentage of people died from the surgery? Well, 1 out of 10, 10%, and that was in the results section. So anybody want to suggest how this study could be improved? I'm looking for answers, guys.
Randomize.
Yeah, how about your placebo, real placebo, sham surgery, blinding, you want a double blind, randomized. You want some safety measures, right? We spent a lot of time talking about the FDA in our group and the fact that they want to keep folks safe. And they would never let a study go forward. You know, they review a lot of studies when you start to plan a study in the United States. Pretty typically the company will sit down with the FDA and say, how does this design look? And if you present a design that doesn't have some safety measures in it, the FDA is going to say, you got to have some labs, you got to collect safety data, because our first step in developing a drug when we're transitioning to people is what we call a phase 1 trial. Phase 1 trials are really safety. They're really safety and a little bit of pharmacokinetics, but they want to make sure the first time you give it to a person, you don't do real harm. So, this is a critical part of trial design.
And the IRB board, Terry, the IRB board that approved that design didn't do a good job either.
Thank you, Ristalina. Safety, we talked about that yesterday, one of the reasons for informed consent, and one of the elements of informed consent is to tell you what the risks are. And so, this is really important. And also, how was the trial conducted? Were there any major protocol deviations? We had no information about that. We need to know about that. What was the protocol? Did they stick to it?
I have a question. If you could just go back to that. The first one where you said placebo controlled and you said a sham surgery, were you suggesting that they actually drill a hole in someone's head and put nothing in there, or were you suggesting like a fake bandage?
No, it would be a saline. What do you know that they did that in the neuron trial, the stem cell trial? Mm-hmm. Everybody got a bone marrow, whether they were going to get treated or not. Everybody got a spinal tap.
Okay.
We mentioned in our group that, early studies in Parkinson's disease for implanting cells, that the ones who they drilled a hole in actually got better because they were getting better from the reaction of the brain to the hole, not to what was being implanted.
Oh,
So
wow.
it's an important, and in fact, we actually got a research model out of that, because not only was it the reaction of the brain to those cells, but they figured out that in animals, if they did that, if they put a hole in these animals that had a Parkinson's disease model, and then just put some gelatin in that hole and then woke the animals up and let them go for a while, and then went and took that gelatin out later on, that they could grow those cells in the lab and learn about this disease and learn about Parkinson's disease. So, so those sorts of placebos are really important.
Mm-hmm.
Can I ask a question about randomization? So obviously the neurone trial, what we do know, they talked about the placebo group being much healthier with an ALS FRSR at baseline than the non-placebo group. So the treatment group, how do you guys, like, what should have been done differently with the trial design for that? Like, do you randomize within, 40 to 48 and 35 to 40, or what's the best way to randomize to eliminate that imbalance in the placebo group?
So two, one, having, we achieve that partly by having large sample sizes. So if you have a bigger and bigger group, it's less and less likely that you're going to have these sort of chance imbalances. If there are features that you think are strongly predictive of the outcome, like your baseline ALS FRSR or the rate of progression up to that baseline, you can what's called stratify your randomization, so that just as you're saying, you would bin people into the 40 to 48 and the 32 to 40, and just randomize within each of those separately so that you ensure that there's balance at baseline for that characteristic. You can only stratify those so many characteristics. It's actually there are newer techniques called re-randomization, where you keep randomizing until you get to something that seems balanced, but that's hard to do while you're recruiting over time. If recruitment takes 6 months, then you don't know who that 6-month person is going to be when you're randomizing the first one. There are minimization techniques. Anyhow, there's a whole literature on different ways, but it's a concern, and we usually use stratification for one or two characteristics to try to ensure balance.
Very good. So our final lessons really have to be: read carefully. Don't take everything at face value. Be informed. Educate others, right? You want to take this information that you're getting in the CRLI to others. And so you don't take everything at face value. You really read critically. And thank you for being a research ambassador and taking this on. We appreciate that. And I think now, any questions or any comments? Do you feel more like you could read a paper and not be so scared and intimidated? I hope so. I hope so.
Honestly, that extra knowledge puts more pressure on, more you know, more pressure to be good at it.
Well, I'm sorry.
I know, it's all good. I mean, it's a rhetorical kind of, very informative.
Well, we hope that you'll at least think when you read a paper now, know that there are other things, that you shouldn't just take it at face value. That alone, take that away alone. And so
I want to reemphasize Rick's point too. If you read a paper and you think, I don't know what I should think about this, write to one of us and say, I just read this paper. What do you think about, maybe they did a mixed model, repeated measures, blah, blah. What the hell is that? I don't know what this is. Is it account for missingness? And I can help describe it for you.
Let me say that if it's statistics, please email Eric.
Well, and then wait a little bit for me to on how many emails are coming in.
Oh my goodness. Well, we're going to move on now. Is there any other comments anybody would like to make before we move on and really get into the nitty-gritty of what kinds of things can you do to be advocates? Well, very good. Nobody else has any comments. Everybody's
I
tired.
just did have one quick comment. I really want to thank you because every time I saw something published, you know, especially if it was a peer-reviewed journal, I kind of thought, all right, I don't really have to look at that so critically. And this really showed me that I do. So this was so valuable to me.
Thank you, Deb. So let's, we're going to talk a little bit about what opportunities there are and how we can be advocates. And Lyle's going to kick it off and with talking about some what the congressionally directed medical research program is and about consumer reviewers and how you can be active. Go ahead, Lyle.
Sure. So, and the first thing I want to say is, so this is going to be pretty brief. I'm just going to talk for like 10 minutes because there's more important talks coming after me. But Rick mentioned the Peace Committee meetings, and in June, I'm giving a longer talk to the Peace Committee Zoom about this program. And so please come and join and ask questions. And a big part of that presentation is going to be some ideas we have about how we can institute a training curriculum to help train consumer reviewers to evaluate research proposals. So much like Terry was just taking us through evaluating a paper, you know, what's the process look like for evaluating a proposal, and how do you consider which ones you should fund? So I'm a volunteer for this, right? I'm not an employee of the DoD or the CDMRP. This is a volunteer job, but it's one that I'm very grateful for the opportunity to do. And I owe that, as you'll see in a moment, to advocates and to consumers. And the reason I'm grateful is that I really think that what we're doing is making a difference, and I wouldn't be in that position otherwise. There's a website you can go to that has a whole lot of great information. If you just Google ALSRP, there's a brand new program book on the website which talks about all the programs and the way that patients and caregivers, what the DoD calls consumers, are integrated into everything. There's, we sent out a big survey about what does the community want us to be doing. There's information about that, other sorts of news. We have this resource database now that's about to be majorly updated that has lists of all these publicly, openly available resources for research. So, what is this thing? So the CDMRP is part of the Department of Defense. It's under the Army Medical Research and Development Command, and it only exists because of grassroots, advocacy. And in the 90s, this huge grassroots effort actually in breast cancer got this thing created. And breast cancer was the first CDMRP program. And when they started it up, they said, we don't want to reinvent the wheel. Let's figure out how to do this. And so they talked to the National Academy of Medicine, who said, you should do a 2-tier review. And we'll talk about what that is in a moment. And importantly, they said, you need to fully integrate consumers, patients, caregivers, people who are involved with this disease into everything, into your investment strategy, into your research focus, into your policies. And so from day one, the CDMRPs, of which the ALS research program is one, this has been the marching orders. And the ALS research program was started in 2007 and has grown since then. And there's a bunch of other ones too. So, looking as a whole at the CDMRP, as I said, this started from a grassroots effort, and there have been over 2,000 consumers representing over 1,000 different organizations that have served on the peer review panels and the programmatic review panels. These are the two phases of review for these programs. And so these are all the programs right now. That ALS is there. You'll see our budget for this year is 40 million. It's bigger than a lot, smaller than some. Breast cancer is 150, prostate cancer is 110. And ours was much smaller than this up until a couple of years ago. And it's only because of some wonderful advocacy that we've been able to have our budget increase. And this is showing that, right? So I joined this committee back in the programmatic panel back in 2013. And I'll tell you, this funding level, 7.5 million sounds like a lot, but the funding rate was so low that ALS scientists didn't really want to apply to this because they said, why the hell am I applying? You get 200 applications and you fund 2. So it's just a game, right? And, we worked really hard to refine our mechanisms and really do things that had an impact and fill a niche in the funding that wasn't otherwise being filled. And we were successful. And because we were successful and we could show metrics of success, our funding has increased. and really increased rather dramatically lately. And so now we're able to fund a much higher rate than we used to. You know, this is total number of grants funded across several mechanisms, and it really is something where now it's much more meaningful than it ever was before. So in last year, our budget increased to 20 million. That was a huge step up, and it's because of advocacy and because of, you know, the Congressional ALS Caucus, and when we kind of knew that this was going to happen. Just real quickly, sorry, I just cobbled these together because it wasn't what I was planning exactly to talk about. Check out the book. This is basically our marching orders. And so everything we do, we're not just looking at basic science. We're not looking at things, you know, this is every funding mechanism we have is designed to accelerate therapeutic development. Through FY19, we only had 2 mechanisms. And one of them was this Therapeutic Idea Award. And the idea was that this is early stage, new, cool ideas, high risk, high reward, right? Stuff that may completely flame out and do nothing, but stuff that could be really, really a new therapeutic, you know, mechanism, a new thing to treat patients. And then the other award was to fund stuff that Jinsey talked about yesterday, kind of something that's more developed, but there's things that you need to do to get it to a clinical trial. You have to do sorts of chemistry and manufacturing practices and, you know, toxicity studies, what are called IND enabling studies. And that's something that has always been underfunded in this space. And you know, a lot of really great ideas have kind of not gone forward from academia because of the lack of that funding. So these were the 2 mechanisms that we had prior to the last couple of years. I'm not going to go through this, but the sorts of things that they were funding. So new drugs, new models to use in the lab, ways to deliver drugs, studying pharmacology of these drugs that were coming into trials. This is just some of the success stories or ongoing success stories of this program and the therapies that it's been developing over the past few years. And so when we learned that we were going to have this increase in our funds, you know, we don't want to be like a room of 8 people making decisions. We wanted to crowdsource the combined wisdom of everybody, of advocates, of, you know, philanthropic organizations, of research scientists. And so we sent out this very detailed survey to whomever wanted to respond. And the survey was to figure out, do people actually understand what we do, right? What are the mechanisms? How do we evaluate them? How can we improve them? Are there other things that we should be focusing on? And so, you know, 3 of the questions are listed there in the bottom right. And what we heard back from, and by the way, that plot kind of shows who responded. And so patients and caregivers were a big number of the information we got back. And what we heard was that people really wanted us to prioritize this mechanism for early stage new ideas, right? Not the same old stuff, not repurposing something, not whatever, but, you know, really novel, innovative ideas that we're having a hard time getting funded otherwise. That there was a real push to emphasize biomarkers and to emphasize open data sharing, right? And the idea that anything that this is funding, that the results and the analysis and the resources should be easily and openly available. And so that's what we did. So we, in FY20, we, number one, we added pharmaceutical industry representation to our panel because we wanted to hear from them, how can we help de-risk the trials? And what we heard was, you need biomarkers. You need ways for us to know that the drugs are working because we don't want to pay for big trials that fail, right? We integrated into the awards based on those responses, you know, requirements, and people have to share their data. They have to share their resources. We added a new award mechanism. And actually, I think Terry was the one who had first sort of championed this because Terry's on the programmatic panel. And the award mechanism, well, the award mechanism in general is to leverage these publicly available resources, but Terry's thing that she really added to this and that really has kind of taken off is this idea that we should be looking at ways to optimize components of current ALS care, right? So the stuff we talk about, the stuff that we currently do that we know works, we don't really have good studies or good ways to know how to use it best, right? When do you start these things? How do you adjust them? And we need more of that. So, you know, we kept those 2 awards. We modified them based on what we heard in those ways. We added this new award in FY20. And the other thing that this award could do is if somebody had an ongoing clinical trial, this award could add an aim to collect biosamples, right? We've talked about some of these recent trials and you know, how we don't know why they fail when they fail. So this award lets somebody fund something to say collect blood or collect other markers from a trial that they already have funded. That new award mechanism in its first year, we got all sorts of stuff back, right? People looking at optimizing, ways eye communication, robotic bracing, new disease progression scales, really neat response. And then this year, we actually had our budget double again to 40 million. And because we have all of these therapies that are in these early stages of development, we are kind of uniquely positioned to now move some of this stuff forward and really into early phase clinical trials. You guys heard about the platform trial yesterday, right? We want to get stuff to the platform trial. We want to have a menu that the platform trial can pick from of really amazing, promising treatments that aren't just the things that somebody else has decided, oh, I've got a drug and I'd like to fund, that really are the good ideas. And so, I showed you some of those other programs in the CDMRP. Some of those larger ones have been doing clinical trials for a long time. And so we talked to all of them. We surveyed all of the different disease CDMRPs for all of their mechanisms. We said, hey, what's worked for you? What hasn't worked? What things that you've done for clinical trials have helped move drugs forward? Can we adopt this for ALS? Can we find parts of what you've done and make it work for us? And based on that, we were ready to go when this award with this increased funding was announced, with a very developed mechanism to fund early phase clinical trials that are dependent on biomarkers, right? And really designed to de-risk larger phase trials into the future. We also increased the amount of that clinical development award for this coming year. And importantly too, hearing back from the community, we have set aside funding to help fund research that was impacted by the pandemic, right? That really was not able to be done because of what's happened this past year. And I'll tell you, this increase in funding for these governmental programs, I'm hoping is going to help fill a gap that's been created by some of the foundation and philanthropy related funding sources that are suffering. I'm not going to, I've already talked too much, but these are our mechanisms for right now. And right now, where we are in this process is that we've just gotten these early letters of intent that we're about to review. I wanted to just spend a couple minutes talking about how the review works. So we have this two-tiered review, peer review, programmatic review, two separate panels. The first tier is peer review, and it is an evaluation of these grant proposals based on their technical merit, based on their science. It's a very detailed review. The panel is comprised of scientists and consumer reviewers, and you guys are on equal standing and you serve similar roles, and everybody reviews everything and debates it and scores these things based on the criteria of the award mechanisms. And there's a training process to teach how to do that. And then the outcome of that is summary statements from that panel of each award and how well they fit the award mechanisms. And then the next step is that it comes to the programmatic review. And programmatic review is a separate panel. It's a standing panel. Again, consumers, clinicians, and researchers. And this one doesn't change every year. This one is standing members, usually for several years. And at this stage, we're doing a comparison-based evaluation. So we're looking at our portfolio. We're looking at things that we want to focus on. We're looking at having balance. What's the potential for impact? It's not individually evaluating each award or re-evaluating them, but rather comparing them to each other. And then based on that, funding recommendations are made. And so this is our programmatic panel for this year. And Terry and I are both on the panel. I don't know who here might know Nadia, but she's just joined our panel as one of our consumer members. The other consumer member is Fred Carlson, who's treated at the Packard Center at Hopkins. And we have representation from a bunch of other foundations, which is useful too, because having NIH and the ALS Association in the room means that we know when they're funding the same stuff, and we can make sure that there's no duplication and that that we're really having the best impact. And I'm going to skip through this. And so where are we right now in this process? The programmatic panel, including the consumer reviewers, is involved in vision setting and deciding what we do, how we make our mechanisms, is involved in what's called pre-application screening, which is what's going on right now, and then is involved in that programmatic review. That's separate from the peer review. And you're going to hear next from somebody who I think served as a peer reviewer last year and their experiences. And I'm actually going to not talk anymore. I'll just mention that the pre-application screening is something that we do extra, even beyond those 2 rounds. And it's basically what's happening right now. Terry, you're about to get all your pre-apps next week, by the way. And it's a way for researchers to submit a smaller summary that gets screened. How well do they fit the programmatic ideas before that first round? And so the thing that I just wanted to mention that I'm actually going to talk about to the Peace Committee is that we want to make sure that we're doing everything we can to let you guys not just learn about the program, but learn how to be reviewers, right? And so we're going to start a curriculum basically that we hope is going to be teaching you how to be the best peer reviewers you can possibly be. And how do you read a grant proposal, and what are you looking for, and how do you evaluate them, constructively in a way that helps us choose what are the best ones to fund. And so stay tuned for that. It's something that I'll be talking about to the Peace Committee.
Thank you so much, Lyle. So, this is an opportunity for those of you who are scientifically bent or would be interested to become a reviewer, either in the programmatic review, or we're going to hear from Todd. He was actually part of a peer review group, that first step, getting past, getting scientific merit that then goes to the programmatic review.
I just want to add one thing I should say. So our goal is to have a training curriculum, and some of the other CDMRPs have done this. It's not something that would be required, like you need to do this to be a peer reviewer, but it is something that we would keep track of who has done it and the pool of people that are being picked from, to be reviewers. And the only goal is to do whatever we can to help you guys, fill your role and really help us. So.
Very good. So Todd is going to tell us a little bit about his experience as a consumer advocate reviewer. Todd?
Good morning, everyone. I'm Todd Kelly. And just to explain before I talk about that is I'd like to let you know that when I was diagnosed in March of 2013 with ALS, I didn't get into advocacy immediately. I mean, when you're diagnosed, as many of you may know, there's a flurry of activity that has to go on. I mean, at the time my two sons were in college, my daughter was in junior high. We realized my wife needed to get a job that was going to have some kind of medical benefits since I would be losing my job eventually. So she went back to full-time school to get a nursing degree. Other things happened, like, for example, our community got together around here and they surprised me with a GoFundMe page, and it was a bucket list trip to England and Ireland for my whole family, which was wonderful, which was set up. And then also there are things I had to do. As my legs got weaker, I had to take driving lessons to learn how to use hand controls, and OVR sponsored that. The Team Gleason sponsored me so that I could do voice banking, so I could use Model Talker. And then I also kept teaching. I directed many plays for about 4 more years, and I was able to teach a full 6 years beyond my diagnosis. So there was a lot happening, and I really couldn't get too involved in advocacy. I did some things. Terry will remember in 2014, we did the Season of Giving letter with my family. I also worked with a videographer, Tom Camano, who was doing a documentary about the ALS Hope Foundation and about Dr. Heiman-Patterson. I spoke at the ALS Hope Foundation Gala in 2018 and various other things. So that's where I'm coming from. Finally, in 2019, in the fall, it was, I think, August, my wife and I had this CRLI training, and it gave me an opportunity to start thinking about how I could do more for ALS advocacy. My progression, as you can tell, has been relatively slow, and there's many advantages about that. Obviously, I've had time to adapt and adjust, but the negative side is, yesterday, I know you talked a lot about clinical trials. I couldn't get into any clinical trials because my progression was slow. They didn't want me. They want people whose progression is going very quickly so they can see a change in whatever treatment they're trying. So I wasn't eligible. And after 3 years, really, they don't want you into trials as well. So I took this course with my wife, We were so excited about all the cool ideas we could do here. And we were looking for just the right thing. And then the pandemic hit. And there weren't a lot of opportunities. Now, Neil's, Dr. Bedlack's organization, does a great job sending out information. They send out listings of clinical trials. They do meetings, webinars, other information. And it was near the end of May last year when I got a particular email from Neil's. And I'm going to try to pull it up here. I'm not used to this share the screen option. So give me a second here. Give me 1 second here. Let's see if this works. Did my screen come up? There we go. Can everyone see that?
No, it didn't come up.
Hit share, Todd. There's a
there?
button that says share. Hit the
Oh,
share.
the button says share.
The green.
Middle bottom, green
Middle
button.
bottom. I'm looking.
You may have to hover your pointer near the bottom of the screen. There should be a green share screen.
I'm clicking on that right now.
Okay, and then it pops up a window.
Yes, it pops up a window.
And then you can
let's
choose
see.
to do it
Here we
one
go.
window or the whole
This
screen.
one. Hold control. Okay, no, I just want that one. Okay, here we go. Hold on.
Yep.
Okay. It's a good thing I'm not teaching right now because I never learned all these Zoom things. Here's what came across, and I got really excited about this because it was something I was looking for. And this is what Dr. Ostrow was just talking about. Individuals living with ALS or their caregivers, your input is needed to help review biomedical research proposals. General Dynamics Information Technology, GDIT, is contracted by the Department of Defense Congressionally Directed Medical Research Programs, CDMRP, to recruit individuals living with ALS or their caregivers to serve as consumer reviewers for the ALS Research Program, ALSRP. Man, that is a scary lot of abbreviations there. But despite that, I went on. Consumer reviewers serve on panels reviewing biomedical grant applications to support the development of highly innovative and impactful research. Consumer reviewers act as lay experts on ALS and bring their experience and perspectives to the evaluation of medical research applications. They represent the ALS community and provide crucial input to the panel, serving alongside scientific and medical experts to represent those most directly affected by the disease. And then the last part was kind of nice. Consumer reviewers are engaged by GDIT as consultants and are compensated by GDIT. So, hey, some food money there. So that's good. How do I get rid of this then?
End share.
End share.
In the bottom right, if you hover over the bottom right, there's
that one's on the top.
Could be anywhere, right? It could
Mine's
be
up
anywhere.
top right now.
Yeah, I'm sorry. Yeah,
Not share. There we go. All right, am I back?
Yep.
Okay, good. There we go. All right. So, despite the preponderance of scary abbreviations, I decided I really think this was something I'd like to learn more about. And so then I went to that website that Dr. Ostrow shared, and I found out all kinds of things. I found out, about how it started for ALS in 2007 and how eventually it increased. And like we found out, it's been increased again to $40 million for this upcoming year, which is a great deal for ALS research. I know it's kind of a drop in the bucket for the government. I guess I should say, as a bad pun, a drop in the ALS ice bucket, but it really is a substantial increase that's really helped. I found out that as a consumer reviewer, I would have a little more than a month to read several scientific research proposals. I would write short reviews for each and provide an initial score. Later, I would read the reviews of scientists and clinicians and have the opportunity to revise my review and score. Ultimately, the entire panel would meet in September for 2 days to discuss all the proposals and provide final scores. Okay, reading, writing, discussion. I was a high school English teacher for 35 years. This is exactly what I always did. So I said, this is great. This is a thing of advocacy I can do. This is in my wheelhouse. So I mulled it over that night. And the next morning when I got up, I said to my wife, I've been reading about this thing. I want to do it. Can I go for it? And she said, sure. All right. So that seems simple enough, but there's actually a process to get into this. It was like getting a job. I had to write a resume. I hadn't done that in 20 some years. I had to write responses to written questions. I had to do a phone interview. I needed a letter of recommendation from someone in the ALS community. So of course, Dr. Terry Heiman-Patterson wrote that for the ALS Hope Foundation for me. So after all of that, I got the gig. It's really a fantastic program, folks. And if it's something you think you can do, or it's something you think you might want to try to do, I would highly recommend it. I met with a number of the other consumer reviewers. These are pals and cows. Most of the people were pals, and there were different people at different parts of the progression. Some of them clearly were having difficulties with speaking, yet they were on the panel. There was one member of the panel who used a computer voice interface so that he could still be part of a panel. You could even, they were very inclusive. If you couldn't type, you could have someone come on as long as they shared the same confidentiality agreement and type the things for you. As a consumer reviewer, I was to focus on two parts of the applications, on what's called the lay abstract, where supposedly the scientists say it in a simple way that anyone can understand, and the impact statement. Although in each case I was given the full research report. And I found that helpful because sometimes I did have to go back into that to understand what was going on. I used to think that English teachers were verbose, but having read some of these scientific journals and these scientific research projects, they're much worse, trust me. I don't think I'm giving anything away here, but in reading one, they talked about testing this on, excuse me. They talked about testing this product on, and then this list of like 8 words that made this gigantic phrase. And I kept thinking, what are they talking about? What are they talking about? And after about 5 minutes, I figured out it was monkeys. That's what they wanted to say. They were testing it on monkeys. But they couldn't say monkeys. They'd use 8 words. I learned new vocabulary. I have never used the word emesis in my life. But now I know it's a nice fancy way of saying vomiting, but I had never heard that in all my years of teaching. So I learned some things that way. The other thing I found kind of confusing for me were all the abbreviations and acronyms. There's usually a glossary, a list. I'm used to that from education, but they're different. For example, I didn't know what GLP meant. GLP, I didn't know what it was. And then I looked it up and I found it meant good laboratory practices. And that made a lot of sense. Throughout the process, I got the support of my SRO. That's called your scientific research officer. Well, I came from a high school. In high school, we have an SRO. That's your student resource officer. That's the cop in the school. So every time I kept talking to my SRO, I keep thinking, this person's not a cop. I was a little confused there. And then, I mean, it works the other way too. I was, like I said, I was an English teacher for years. About a year after I was diagnosed, I was the head of the department. I had to send out a notice about this new program we had, which was called Alternative Learning Strategies. And someone in education thought it would be really cool to abbreviate that as ALS. Bad choice. Half my department wouldn't open the document because they thought I sent them something that I meant to send to my doctor by mistake. So, you know, I don't know who makes these choices, but these are some things I learned. The recruiter who got me involved in this cautioned me that there was going to be a lot of reading involved with this. But trust me, as a high school English teacher. And also I read for the College Board with AP, where we would read thousands of student essays for 7 days straight, 8 hours a day. This was nothing compared to that. All right, so I did the reading, did the work, was ready for the 2-day session. This was in September. Now, normally we would all meet down near Washington, somewhere down there. But of course, wonderful year, we decided we'd have to do it through Zoom, just like this meeting. I mean, you know, there are certain advantages. Number one, you know, there's no travel involved, which is very convenient for someone with ALS, you know, because I have problems with mobility. You know, easy bathroom facilities because I'm at home. Good work area set up. I had 2 computers set up. I had papers. I had my cup of coffee. I was ready to go. It was great. But there were disadvantages, of course, to Zoom. And you found that out too. You don't get the nice meals. You know, that's always good. And what I really missed was the social networking time, talking with the people when you weren't just focused on what was going on. So I did miss that greatly. I'm glad that Terry here has these like breakout sessions. I guess you had a social hour last night. That was good because I find that's very valuable. And the other thing was just like now, when we actually did the presentations, you guys are all very polite and you've all muted yourselves very nicely. But when I say something, I'm a high school English teacher. I expect a reaction in some way. I mean, I always considered my 50-minute class kind of like a stand-up routine. Not that it was funny, but that I would keep my audience involved and I could tell if kids were learning or not. See, if I say something funny now and you think it's funny, I have to look at your pictures to see if you're actually laughing or groaning. Or, you know, if I say something incredibly profound, I expect everyone to be like nodding along. So that was one thing that made it difficult. It also led to some odd things. Again, I'm not being specific here because of confidentiality, but the one reviewer kept coughing when he would talk to us. And we found out it was because in his office where he was, the wildfires that were going on last year at this time, the smoke was coming into his room and he was having trouble with that. In fact, we were a little concerned. We said, are you sure you should be there? You know, maybe you want to move, but it was supposedly everywhere along. Okay, so as I said, there are strict confidentiality agreements. There's proprietary information, so I can't really share a lot of specifics, but I want you to know it was an incredibly valuable and inspiring experience. I felt that the input that I provided was sincerely valued by highly qualified scientists and clinicians. If I had a question and I asked it, they were more than willing to answer it. I learned so much more about ALS. And I, you know, as a teacher, educator, I'm also a lifelong student. And I love to learn. And the more I learned, the more I feel confident knowing that what's being done or what's attempting to be done for this makes me feel better as a PALS. I probably learned more about the SOD1 mouse than any human person needs to know, or the SOD1 mouse, although I'm sure Terry knows a lot more than I do about that. But the other stuff I learned was incredibly valuable. Because of the confidentiality, that was the one drawback, you know, because I learned some wonderful things. I would have loved to, you know, talk with Terry about those afterwards, but I can't. But I wanted to let you know that my score did count just like everyone else's. And I found that wonderful. We were able to fully discuss and score all the proposals in a 2-day period. Oh, by the way, the other thing about Zoom that's annoying is right now I've got a cat here who wants my attention because this is his time to sit in my lap. So in a moment, if he leaps up here, you'll forgive me. With these projects, because of the time, I realized that maybe some of the things I was reviewing, probably most of them, would never really impact me. But I was hoping that I could play some small part in helping future PALS and encouraging this invaluable research. I kept that constantly in mind, that I needed to represent all people with ALS. For example, if a study was just designed to help familial ALS patients, you know, about 10% of the population, I don't have that as sporadic, that wasn't something for me to consider. I wanted to help ALS patients and speak for all of them. So that was just as valuable. And besides, anything that would help there would probably come back to help sporadic as well. Although this panel is not for everyone, I anticipate with the increase in funding that Dr. Ostrow talked about, going up to $40 million, they're going to need more people. And again, I liked your idea of maybe having some kind of training session to help with that. Luckily, having been on many evaluative committees in the past because of my work as a teacher and, like I said, for the College Board, it wasn't difficult for me to kind of adjust to this because I've done this type of work before. My final thoughts here, my primary message to anyone who's listening right now, anyone who's becoming an ALS ambassador, what you need to do is find the vehicle that works for you and your particular skill set and use that. If fundraising is your forte, then go for it. If you can organize a 5K, great. If you want to coordinate and attend a political rally, pursue that. My experience as a high school English teacher and AP reader made becoming a consumer reviewer a logical and personally rewarding means for me to contribute to the ALS community. So those are my thoughts. If you have any questions, I'd be glad to answer them.
Todd, I just want to say, as a fellow pal, dude, you're inspirational as all get out. I mean, I'm sure you've heard that. I'm sure you've read Viktor Frankl's Man's Search for Meaning. And his basic premise is if you know your why in life, you can survive almost any what. And so clearly you have found your why in life. Why you're alive, why you're doing what you do. So I just want to say thanks for sharing. I will think about a lot that we talked about this weekend, but I will remember you. And I appreciate that. Thank you.
Thank you very much. And that's the tough thing with ALS, because it makes you question your why and your what.
Right.
And what, because I love being a high school English teacher. And when this happened and I realized that that was not going to be necessarily something I could continue, that caused a lot of, personal searching for that purpose, that meaning.
Yeah, and that's clear. I mean, you exude that. So it's awesome. And thank you for taking the time to put together such a great presentation. So thank you.
Thanks.
Todd, you're just the gift that keeps giving. You are terrific. I can't even. Let's give him a round of applause, everybody.
And Todd, I have one question, and it's you just talked about how much work this was, and it is work, and I'm grateful. I'd like to request a little more work. Can I reach out to you separately to talk and ask you some specific questions about your experience? Because we really, we're trying very hard to design a curriculum to help make this better, not just the training part I talked about, but for example, you mentioned the impact statements. We want to revise instructions we give to researchers so that they're writing impact statements that are more helpful to you. And, so we need your insight to help figure out what those things might be.
Okay.
Is that okay? Do you mind? I need to spitball some stuff.
Okay, no problem.
That was just unbelievable, Todd. Thank you. So we're going to move on, and let's see. Jamie, would you and Gary like to move forward? Go ahead and start.
Okay.
Jamie's going to talk about some experiences he's had in group advocacy along with Gary, but go ahead, Jamie.
Well, I'm going to talk about an experience I just had. Todd, that was wonderful. And I thank you. And I want to share something with everybody. I'm actually emotional now, Todd, because you remind me of this guy, my best friend, who was a teacher, and he used his voice to make a difference like you, Todd, and for all the patients that are here today. Thank you. You keep teaching me, you're teaching me to continue his legacy and the passion and why I do this every day. I didn't think I was going to be emotional. I'm sorry, I had something else planned. I was going to tell Dr. Macklin a joke to get back at him for teasing me earlier, but Thank you, Todd. And thank you, patients, for being here and taking the time this weekend to do something that's so important. And I appreciate you all. I'm trying to compose myself now. Wow. So this is Wes. This is why I do it. See, Wes was a patient and he was huge in research and advocacy, and he was part of the one of the initial CRLIs. And he was my best friend. I got to tag along with him and convince him to go out and have a beer and sandwich afterwards. And that was my fun. But he passed away, and I promised him I would continue his legacy, and I would continue his voice and continue to create awareness. I didn't know how I would do it. I'm not as smart as someone like Todd or Brian or Todd here, the teachers in the group, but I knew that I have a voice and I like to talk and tell apparently bad jokes. But how would I do that? And I would continue to go to advocacy days. I would continue to be a team captain for his walk. I would join committees and either caregivers, because I felt like I was a caregiver to him, and also be and join groups. And even today, joining groups like I Am ALS and Everything ALS, who are doing amazing things and starting caregiver programs. I know that I can carry not only his voice, but the voice of so many other patients and families that have been affected by this cruel disease. And I just wanted to make sure I continue to stay involved. And I kind of like what you said, Todd, you find your niche. There's a lot of ways I can't do a paper, but my voice could still be heard, and I can still rally to have people to get involved and write a letter to the congressman or their elected official. And it doesn't matter what you do. We just have to keep moving that needle forward and try to find a solution or a cure for this, because this is what we do. And this is why we do it. And I'm so grateful for everybody being on here. And I just want to thank you. And there's a person here I want to introduce is Gary. And, you know, I was drawn to Gary. I saw a lot of passion in him. And he came to us and wanted to do a 5K. And I got to know him. And the more I got to talk to him, the more I was, impressed by him. And I kind of want to introduce Gary Curtis, because he has a story that I feel is the same. And I appreciate everybody putting up with my tears, but I'm just so inspired. I'm so inspired today just to go out. But anyway, now I'm babbling and I'm sorry. Gary Curtis.
Well, now you've made it almost impossible. How do I follow that? Well, very quick introduction. So I spent many, many years in Maryland. I've worked in DC. I was there, unfortunately, at 9/11. And then 1 year later, September 11th, 2002, I'm going to work and I'm thinking, boy, this is going to be a lot different than a year ago, where I was literally in the city feeling that the city's under attack. And I knew that this would be a different day. 10 o'clock in the morning, I got a phone call, and I'm sorry, I'm getting emotional, but that was the message that I never wanted to hear, that my dad had passed. He went down very quickly, and I wanted so much to do something, but I didn't know what to do. And the only thing I could do is, living in Maryland, the Packard Center was running these 5Ks called the Fiesta 5K. So I ran that many, many years. It was a great run, a lot of fun, very successful. Fast forward, I moved to Philadelphia 4 years ago. And I come to find out that there is nothing like the Fiesta 5K around here. So I started asking around, and I really couldn't get anybody that showed any interest. So then, by pure coincidence, in 2018, I see a sign near where I live in Ambler that there's this walk for ALS. Oh, that's great. So I do the walk, and then I talked after the walk. I had my dog with me, and I talked to a couple of people that were involved with the walk. And I said, geez, you know, my dream would be to have a 5K here to raise money for ALS, because I think that would be a great idea. And they were all over it. So that really started my involvement with the ALS Hope Foundation back in 2018. And that ultimately led to the inaugural 5K that we had, which we call the Run for Hope, which we had in June of 2019. I remember our goal was to raise 5,000. I think, Jamie, we ended up with about 27,000. And that really began the beginning of my association with the ALS Hope Foundation. They since invited me to join their leadership board, as it's called now, where we plan all the activities and so forth. And it's been great. I just want to talk about my definition, which might be a little bit different, of what advocacy really means. And I think, Todd, you kind of touched on it too. To me, advocacy, it's a combination of three things. It's about raising money, funding for ALS, but it's also about raising awareness and providing education. Because what I found out is a lot of people have heard about Lou Gehrig and they've heard about some other things. And obviously the Ice Bucket Challenge was great. But there's just so many people that really don't understand this disease the way that they really need to understand the disease. So I really wanted to get as much involved as I could in those three areas. And the ALS Hope Foundation and the people I work with there, that I volunteer with there, made it so easy to do that, because they are just such a great group to be with. They made me feel like family, which has really made it so much easier to do the things that I do. And it really has been so enjoyable. So then at that point, you know, here I am, I'm involved with all the activities, and I got involved with the walk and several of the other things. And then last fall, Jamie approached me and said, we have this national committee called IMALS. Would you like to join that? And I said, I really don't know much about it. I've never done advocacy. And so I joined it. And you know, I like to say that I am part of a team. You know, I don't take any credit for anything I do there, because they are a great, great team. They do such great work. And it's been quite eye-opening for me to really, it's been an education to really understand what advocacy is all about, how difficult it is. It's just amazing to me that a bill was just passed for SSDI, and there shouldn't have even been a need for a bill like that. And to find out that it took years to do that was just amazing. And now we're working on Act for ALS, and I think everybody here, I'm sure, knows what all the benefits would be related to everything that we've been talking about. So it's been great. I'm involved with a lot of the activities there. They've got a lot of activities going on related to Lou Gehrig Day, so I volunteered for that and so forth. So I had all of that going on. And then I thought, well, wait a minute, I also have my other life where I recently retired, and I have my own venture where I do sales coaching and training, because that's been my entire background over the last 40 years. And I thought, geez, I have two passions. Maybe there's a way I can link the two together. So I started to do that. And the first thing I did is I started promoting the fact that I give 10% of anything that I bring in to the ALS Hope Foundation. Then I thought, well, wait a minute, I've got all this networking that I do, particularly over the past year where everything is Zoom. I've got hundreds of people that I've met. Why not get them involved in what I'm doing to educate them and see how they can now get involved with supporting ALS? And they hear about me. I'm on these Zoom calls all day. That's my life. And they hear about me talking about my affiliation with the ALS Hope Foundation. One of them actually said to me, we don't think your initials are GK. We think your initials are ALS, because that's how we know you know. And I've been very successful in getting them involved with my goals to create awareness, to educate them, and hopefully to create funding. And some of the things we've done, which I'm very proud of, we developed a relationship with Investors Bank, which is a local bank that does a lot of support of nonprofits, and we linked them up, and I think that's going to be a great relationship to bring very important critical funding to the foundation. For our events that we have coming up, such as the 5K, we've got someone that does aerial photography. He's volunteered to do aerial photography for the event. We have an event coming up this month with one of the groups we do. They feature a nonprofit. So I'm having them feature the ALS Hope Foundation. It's called Networking for a Cause. And it's a great way to give exposure to nonprofits. So ALS Hope Foundation is going to be featured this month. Yesterday, I was involved with one of my networking groups. They're doing an event for all nonprofits where everybody contributes and a certain percentage goes to nonprofits that they've designated. And I make sure that the ALS Hope Foundation was one of the ones. So you'll be getting a check very soon for a certain percentage of the money that they collected yesterday. From this group, where they've heard me advocate so much about the need to support the Run for Hope, just in a very short period of time, this group has already donated $1,300 towards the run, just from hearing me talk about how important it is to try to support what I'm trying to do. And then we're involved with the Chamber of Commerce. Someone from the Chamber of Commerce in Philadelphia approached me and said, we'd love to have the ALS Hope Foundation join because we think that there's a lot of people in the city that they can meet that would be delighted to eventually help to support them. So I think, Jamie, your plans are to join the Chamber very soon. So these are all things that I've been trying to do to just get the word out over and over and over again about how important it is to understand the impact of ALS, to understand how important it is to support this local organization that does so much for patient care, for research, and for education. And you know, I'm on my way. I made progress, but I got a long way to go. So that's my story. Thank you for listening.
Thank you, Gary. You've done so much. Gary's amazing, by the way. He just takes the ball and runs, but all the pals on this program, you all have the capability of doing this, educating, raising funds for the organization that you're close to. You know, you can help so much just raising awareness about ALS so people know about it and understand it. Don't hide in the shadows. Take every opportunity. Thank you, Gary. You've been just amazing. Jamie, did you want to add anything?
No, I thank you for everybody for listening to my story. And ALS is raw, and it affects so many different ways. And so I appreciate it. Thank you. You're gonna make me start crying again, Terry. But
Jamie, the only thing you left out was the magnet story, of course.
The what?
The magnet story.
Oh, yeah, we'll tell that some other time. We want to get Dr. Bedlock. But I do have one thing, Dr. Macklin. You know, I did have a presentation. So what do you call an annoyed lobster?
I don't know.
A frustration.
A crustacean is frustrating.
All day long, I'm telling you guys, all day long I listen to it. But you know what impressed me, Jamie? I didn't know you owned a suit. I saw you in a suit in that picture.
All right. Clearly, we're back on track.
Wait, I have a quick question. I saw in the chat that Todd might be leaving. So are we going to be able to exchange contacts and everything?
Yes.
Okay.
Yeah, we're part of the family now, Deb. This is this is our group. And,
okay.
So, yep, everybody will have it, and we're going to send that out after the. At the end of the day.
If anybody doesn't want their contact information sent out for whatever reason, please let us know, because otherwise we're going to share that.
Yeah.
Okay. So we're going to finish up this session with Rick, and he's going to tell us a little bit about the Peace Committee and how to sign up with the NEALS tracker.
Yeah, thanks, Terry. Fantastic presentation so far today. So, you know, one of the most common questions that we get in the surveys that we're asking folks to do at the end of the program is, it would be nice to tell us where to go next. Like, we don't know what the next thing that we should do after this program is. And so you heard about some great examples from Todd, Gary, and Jamie. But here's another way for you to regularly get ideas for influencing the research process, for engaging in it. So we've got this committee, which is called PEACE, Patient Education and Advocacy Committee. It's actually one of the subcommittees of the Northeast ALS Consortium, NEALS. And you can see our mission statement there. We want to utilize education and advocacy to empower persons with ALS and their families to have a greater understanding of and a greater role in clinical research. How do we do it? Well, we have monthly teleconferences, and we invite different stakeholders, patient advocacy groups, sponsors, and researchers to come on and present projects that they want input from research ambassadors on. And so now that you've graduated from this program, or you will in a few minutes, you're all invited to be part of the PEACE committee. And how do you get on the committee? Well, I believe that Terry is going to submit all of your contact information to the two co-chairs of this committee, Allison Boulat and Nadia Sethi. Is that right, Terry?
Yeah, and we also have, I think, from Carly, a how to sign up sheet that we will share with everybody.
Yeah, to sign up for the PEACE committee, all we have to do is have your email, and then you'll start getting invitations. If you want to go ahead and put some of these dates on your schedule, these are the upcoming PEACE committee meeting dates. If for some reason you can't make one of these, we always send out minutes afterwards so you can hear about the projects that were presented, and you'll have an opportunity, if you missed the call, to engage with that particular stakeholder in their project. Now, of course, the PEACE committee is not the only place where you can get ideas. There's an unlimited number of different places where you can get ideas. And I showed you in the beginning some of the amazing things that some of our research ambassadors have done. But to remind you, two of them went on to start the Ice Bucket Challenge. They've participated in advocacy for different kinds of bills and laws, including Act for ALS. They've been part of the development of the FDA guidelines for ALS research. They've helped us to get the Social Security benefit delay removed. They've started podcasts. There's one out there called I'm Dying to Tell You, another one related to ALS Untangled. There's a group of research ambassadors now that are doing something called research paper reviews. This is kind of like a journal club where they pick out a paper, they ask the authors of the paper to come on and present it, and then they field questions from patients and caregivers about the research. They've been part of advisory boards, as you heard about, and of course they have their own different social media groups. For example, there's several of our ambassadors that have Facebook groups. So what I would ask you to do is to look at your own unique skills, backgrounds, hobbies, and interests. And don't be afraid to find a way to use those in some unique method to raise awareness for ALS. So, thinking about my own journey, I've learned that I'm a good listener, that I'm creative, that I'm not afraid to work hard. And those things have allowed me to recognize a need for and build things like the Duke Multidisciplinary ALS Clinic, ALS Untangled, and this Clinical Research Learning Institute. And if you know something else about me, you know that I've long had a weakness for outrageous clothing. I just find that, it lifts me up. There's days when I'm tired or down, and I've got to find a way to stay energetic and positive for my patients. And the outfits help me do that. And they're also great icebreakers. I know that people come to my clinic and they're afraid of the measurements that we're going to make and the things that we're going to have to talk about. And so if the first thing they see is some kind of crazy outfit, I think it puts a smile on my patients' faces, and it's a great way to start the day. And so this year during the pandemic, I started to look at my own bucket list. There's only a few things left on there, and I decided to put all these things together and accomplish one of my bucket list items. And that was my first custom-made suit. So I'm not going to spend a lot of time talking about this, but this suit is a style of suit called a nudie suit, N-U-D-I-E, because Nudie Cohen in the 1940s was making these embroidered Western suits for country Western musicians. Like you might have heard of someone named Porter Wagner. He had some of these suits. Elvis actually had one of these suits. The suits that he made for those guys were embroidered with symbols of the Wild West. I decided to have my nudie suit embroidered with symbols of lessons that I've learned from people with ALS. And there's a whole story behind every symbol, which they're too long to tell you today. But briefly, on the pant leg, we have motor neurons. Never did I realize how important these tiny cells were to our lives. On the sleeve here, I've got something which is called the staff of Asclepius. Asclepius is the Greek god of healing. And I've learned that even though not yet can I fix ALS, there are a lot of ways that I can heal symptoms in my patients. Here we have the blue cornflower. This is a symbol of resilience. I have found that, like people with ALS, these flowers, when you first meet them, they seem fragile, but they're incredibly tough. And these flowers can maintain their beautiful blue color even through the harshest winters. And I found that to be true of so many of my patients. Up here on the top, we have swallows. I have a whole story about a patient that I met early in my career who taught me how important hope was. At that time in my career, I was mainly focused on the science. What kind of measurements should I be making? What do these measurements mean? What kind of evidence and experience-based options should I be offering based on those measurements? And I had forgotten that probably the single most important thing I do is hope. And so the swallow reminds me that if I haven't said something hopeful to every patient and family, I need to go back in there and do that. And on the back of the suit is a symbol from my favorite TV show, The X-Files. That was from the poster that was over Fox Mulder's desk, said, "I want to believe" on it. And I've just learned, that I've got to keep an open mind. There's a lot of ideas that I've heard that at first seem totally wild, but they just might work. And by keeping an open mind, I've learned about different ways to do research studies. I've learned about treatments that seem kind of crazy, but we need to study them. And I've learned about a group of people who recover from ALS, the ALS reversals that I never knew existed. And so I'm going to continue to remember those lessons for the rest of my career. One of the most important things I want to leave you with today is, I know you're going to go out and do amazing work, but please record what you do on our Neil's outreach counter. So you're going to get the keys to this. It should be in the materials that Terry sent you. Whenever you go out and do some of the amazing advocacy, please go on there and make sure that you click the appropriate column and category here so that we know what you're up to. And the reason, again, this is so important is we're going to have to get funding for future CRLIs. Someday we hope to be doing these in person again, and that gets expensive. They can cost $30,000 or $40,000 depending on where we do them. And whoever we ask to sponsor it is going to ask for evidence that this program is making a difference. And this is one of the pieces of evidence that we need to be able to show them. So that's all I had to talk about today.
Dr. Bedlack.
Yeah.
Can you go back to that picture again?
Which one? This one?
Yeah, one more. Picture of you.
Oh.
That one. I love all the symbols on there, but I found it more striking that your arms are open and accepting to everyone. And you just leave it out there for all of our patients and families. And I love the symbols, but I love the fact that your arms are wide open to accept everyone.
Well, that means a lot to me, Jamie. There's two quotes that have left a huge impression on me. One, early in my career, I heard a patient with ALS say, "Nothing about us should be done without us." And I've tried to make that my mantra whenever I'm on any committee or invited to speak at any meeting. I try to make sure that patients with ALS are going to be part of that. And then the other is Brian Wallach's tweet that he sent very early in his I Am ALS journey, which was "Better Together."
Yeah.
And I just firmly believe that there's far too few people that care about this disease. We should not be fighting amongst each other. We've got to find a way to recognize the good that we all do and, synergize and capitalize on that to move us all forward.
Thank you so much for those words. And I really also just want to comment about you and others on the call. Words are so powerful, and especially if you're a physician or a researcher, I think that sometimes your words hold so much power. So, and I think that you're so cognizant of that, and I just wanted to comment. Thank you.
Thanks, Deb.
Thanks so much, Rick. That was just terrific. And I really do like that suit. Where are you going to wear it?
I'm going to wear it to Philadelphia next year when you and I go shopping at the mall there.
Excellent. I will expect that. We'll even plan a special dinner so that we can go out and you can wear that. And maybe I'll find something matching. All I have is black in my wardrobe. So, you know, it may be difficult, but I'll look for something that matches. But thank you, everybody who participated in this part of the program. And I hope that everybody's getting some ideas on what they can do. We're going to have a little bit of a break. We'll be in breakout rooms, but if you want to go get a cup of coffee, you want to, you know, hit the bathroom, whatever you need to do for a few minutes. And we'll be back at around, you know, after the coffee break, we'll all meet back here. And Dr. Ellman is going to sort of lead us in a discussion and just have everybody sort of think about how they might plan to use this, what they think their skill sets are, what they got out of the 2-day program. You can even just talk about that and how you're going to move forward. And the faculty may have some ideas too, or be able to comment on that. So looking forward to the rest of the morning and enjoy the breakout. And thank you again to everybody who spoke. Todd, unbelievable. Gary, Jamie, you know, tease you, but you're terrific. And, you know, thank you to Lyle and Rick for helping talk about some opportunities and some things that can be done. So let's take our break. Oh, before the break, I think we want to do a photo to tweet out. So if people can turn on their pictures again, that would be great for just a moment. Sarah's gonna. So everybody show us your, if you're able to. Thank you. I don't know if the other folks are hearing us or they're out in.
Already on their break.
You're already on break. Okay.
All right.
Here comes Lauren. Yeah, thanks, Lauren.
All right, cool. We got mostly everyone.
Yeah, I think so. We don't want to hold up break time.
All right, ready? 1, 2, 3. Okay, let me do one more. Ready and go. Thanks, guys.
Okay, so we're gonna take a break, go to breakout rooms and socialize a little bit, and look forward to coming